This is the briefing for estimation of reducing sugar found in banana by dinitrosilicic method. This will be practical too for the biochemistry practical of PSB 1402. Basically, this practical is to estimate the reducing sugar, as you've been well informed in the biochemistry lecture. You will be required to do an experiment where you will rupture the cell and expose its reducing sugar in a banana. And we will choose two types of banana, and we will treat it and expose the content of the cell. We should have a lot of carbohydrate among other things, and some of these carbohydrate could be reducing sugars. These reducing sugars able to be in acyclic and cyclic form, and therefore it mutarotates. And when it's in acyclic form, it will have either aldehyde, carbonyl group, or ketone group, depending on which type of reducing sugar found in the banana itself. And what happens after that, we will then expose it to the DNSA reagent, and it will cause a change in color. The color change or color changes is actually is proportionate. Proportionate means the more the concentration of reducing sugar, the more the color change will take place. And this can be actually quantitatively measured by using a spectrophotometer being set at a certain wavelength. What happens next is you will get the absorbance of this reducing sugar, which can be referred as unknown. And then you will use known, which is standard. In this experiment, we will use glucose as the standard. We will build a standard curve using this glucose at various concentration, and therefore you will get various absorbance, which will be useful in making the standard curve. Next thing, you will use this standard curve to actually determine from the unknowns absorbent to what will be their relative or appropriate concentration by using the standard curve. Nowadays, compared to those days we actually use a graph paper, then get this standard curve, and then by using a ruler extrapolate manually. But nowadays, with the availability of Microsoft Excel sheet, maybe other form of tools there, computer tools, it is possible to just build the graph by using Excel sheet, and followed by using the formula given by the graph, which is y equals mx plus c. You can actually calculate the unknown, the unknown's concentration by knowing the its absorbance. Okay, but before that, the r squared has been written there will decide whether it's a good fit of a graph. The good fit of graph normally will be r squared having close to 1, but not necessarily, completely to be 1. 0.99, 0.98, 0.97 as well can be considered a good fit of a graph. But anything below that, there is something that went wrong in your way of conducting the experiment itself. And then the Beer-Lambert's law also need to be understood. Basically, in brief understanding, Beer-Lambert law suggests that it is the standard curve will be a straight line which passes through the origin. It will be obeying the Beer-Lambert's law. The only exceptions to the Beer-Lambert's law in terms of its limitation, it cannot be useful when at high concentration of the unknown. When at high concentration of unknown, it is not accurate. Therefore, if you want to achieve a better accurate determination of the unknown, you are required to dilute the unknown so that the absorbance is within the standard curves absorbances. Then it is much more accurate. So I hope you find out what is the meaning of this limitation of Beer-Lambert's law without just using a high concentration of your sample. You can basically compare your absorbance. If it is beyond the standard curve's highest absorbance, then better to dilute, repeat the experiment to dilute it. Therefore, the absorbance of your unknown fits within the standard curve. As well as, don't forget, after you get the concentration of your diluted unknown, you need to multiply with the correct dilution factor. So these are all must consider to be accurate in determining the concentration of unknown by using this method. So let's move on. That's on the overview background. We now move to some of the videos that I have carefully selected. I only select videos that are having the highest view, and then I go through it and found to be very much suitable for this practical itself. The first YouTube video that I have given here is actually of my own on how to use the micro pipette. We will be using a lot of measurement of small volumes below 1 ml, which is equivalent to 1000 microliter, or even below zero point 0.1 ml, which is equivalent to 100 microliter. And in some instances, maybe not in this biochemistry laboratory practical, in other application, even below 10 microliter of volume. Therefore, you need to have different micropipettes. But for this purpose, it's sufficient to have that micropipette that can measure below 1000 microliter or 1 ml. And normally the tips for that is blue tips. Okay. And hopefully you watch the micropipette video and therefore you are familiar. Before you come to the laboratory, we will also show, demonstrate hands-on on how to use it. And the next video is on describing the DNSA reaction, a bit on how it works. That's been covered by this video, and also a bit of introduction on the standard curve, the use of standard curve, and related to the related to carbohydrate determination, quantitative reducing sugar quantification. Next video. This one talks about the actual similar experiment that we are doing now, except some variation. We are using wavelength of 510 while they are using 540, but in a way slightly off, but still relevant video for you to watch to understand how this whole experiment is conducted by another person or another student somewhere else. The next video, it is again more on all the step by step guide on the use of the Excel, most importantly, and also a little bit on the description of the experiment as well. So I hope you view all these videos before actually coming to the laboratory, or apart from viewing it before coming to the laboratory, also consider when you make the practical report to have understanding again, revising. Okay. Right. We now, before we go into the experiment procedure itself for this practical, it's best we do some preparations of materials. You have the DNS reagent need to be prepared, and this one could be done by the technical officer in the laboratory, or you also required to have the direct exposure on how to prepare all these materials. We will decide that when we go into the laboratory. But basically, you have to use an analytical balance. You must know how to use the analytical balance using a weighing board. They call it. It's just you know an apparatus or the space to put the material in a accurate or clean way. So you use the weighing board to weigh 1 gram exactly of this DNS. It comes in powder form. It's also good to know whether the material is in powder or in liquid. And then the next one is 200 milligram of crystalline phenol, as well as 50 milligram of sodium sulfide. And then you take all these three material and mix it with 1% NOH. That means this 1% NOH, which is volume over volume. I hope you know what is the meaning of percentage of volume over volume, or weight over volume. So this one is, I'm sorry, it is 1% of weight over volume. So the NOH actually comes in pellets. So it has to be prepared taking 1 gram with 100 ml of water. So that's 1% weight over volume NOH. This one I think is prepared prior by the technical officer. So normally, to be accurate in preparation, let me do a short drawing on it. We use the volumetric flask. I hope you know what is the volumetric flask. It has a meniscus or graduation there, and it has a stopper above. So you to put 1 gram, 200 milligram of crystalline and 50 milligram of sodium sulfide all into the container, okay? And then you drop water, okay, until the meniscus or before the meniscus, because firstly you want to tilt it upside down until it's mixed thoroughly. Then you add further water until it is 100 ml exactly. So that's how you do the preparation of any solution with that involves solid material. Okay. Here it's mentioned if it is to be used immediately, which will be the case for this practical. But if you want to keep this DNS reagent for a longer period of time, it's best to add sodium sulfide. But I think that's not necessary for this practical because we prepare and immediately use it at that instant itself. Next, we have to prepare a 40% Rochelle salt solution. This is need to be done 40 gram mixed with 100 ml of water and also using a volumetric flask of 100 ml. Moving on, we now get into the actual procedure itself. This practical in terms of the procedures are quite, only one experiment, not many steps. Um, but it is, well, I can say it's straightforward. You have to also understand each of the step because you are relatively new for any biochemistry practical. So what you need to do is you have to take the fresh banana, which we will purchase, two types, and then take 1 gram, cut it, remove the skin, only the flesh, weigh 1 gram, and then we add liquid nitrogen. So normally we use a porcelain container. I think we normally use the pestle and mortar, the pestle itself. So we put the banana inside. We add liquid nitrogen. It is very low temperature. So must be careful not to expose it to your own self. If not, they will be harmful. Okay. Normally done by the technical officer. And after that, you have to grind it using a pestle. the mortar, and hopefully it will be crushed and exposing the content of it. And after that, we transfer into a centrifuge tube and added with zero point 0.5 molar of NOH, all done prior to that. Okay, so only 8 ml. And since you are relatively new to biochemistry practical, I also have prepared the YouTube video on what is vortex is all about. This one is just a vortex mixer, which is you press the button, it will create kind of like a whirlwind, swirling kind of ability, potential, and then mix all everything up for you. Okay, so watch the video. And also, maybe you also don't know what is a centrifuge. So I also provide another video. So centrifuge, you have to centrifuge it with the guidance of the technical officer at 5,500 revs per minute. That means spinning speed for 20 minutes. And the centrifuge is actually refrigerated, refer it, and you can set it at 4 degrees Celsius. So there is no, breakdown of your vital reducing sugar. And then you collect what is referred as the supernatant and ensure that you do filtering with filter paper in a funnel. And that also will be shown. The filter paper, usually is Wattman number one. That means very coarse filtration. So it's faster. So you take the liquid out while the sediment is thrown away. The liquid is referred as supernatant, and you neutralize it with some acid. This one, since we want to ensure accurate neutralization, you can actually use a pH meter by taking its probe and, of course, a calibrated pH meter, using the probe to actually mix it and also measure whether it has achieved from alkaline condition to finally to pH of 7 roughly. Don't have to be worried it has gone too low of a pH. Okay, so assuming you have done that, then you pipette this either zero point 0.5, another tube 1 ml, or another tube 1.5 ml, or 2 ml, and another one 2.5. Okay, this one they leave it up to you to decide. Say you have zero point 0.5, another one 1.5, another one 2.5, so 3 tubes only. And every time you take, say, zero point 0.5, you need to add 2.5 ml of water. So it will become a total of 3 ml. If you take 1.5, you add 1.5 ml of water. So the supernatant need to be mixed with water. So total will be 3 ml overall. So here there is actually some dilution. So careful. Later, once you determine the concentration by using the standard curve, don't forget about the dilution factor that need to be multiplied to the answer. Okay. Right, moving on. So once you have, say, 3 tubes, then you add 3 ml of DNS reagent, and then to heat it for a total of 5 minutes using a water bath. So not direct heat like, Bunsen burner, instead using water bath. And then you, when the content of the tube are still warm, so no need to cool it down, just take it out with the test tube holder and then add with 40% Rochelle salt solution, which already been prepared prior. And the next step, you are required to cool down these test tubes before you do the reading of intensity using a spectrophotometer, which has been set at 510. Hopefully, you also know which type of QED to use when the wavelength is 510. Right. Once that is done, the instruction also stated on the number 12 there, run a series of standard using glucose which the stock is 10 milligram per ml. You're supposed to dilute. There's a table below that will indicate how much of concentration that you need to achieve finally for around six test tubes. Let's look at that. It states that after you have done a series of standard using glucose, you are to repeat these steps 8 to 11 again. So let's look at below. Right. We need the standard curve, the graph, in order to calculate your unknown, which is the banana extract, which has reducing sugar. So how do we prepare this standard curve? You firstly need to prepare your stock solution, or maybe the technical officer would have done. We'll wait and see how it goes. And then this table highlights the fact that you need to prepare six test tubes and with this kind of concentration so that you can get a nice graph. And here, not being told how much, so I can just give you a hint that you need to use this formula. Okay, so try to figure out. And actually, it'll be good if when you, before you come to the lab, already calculated all these empty uh, parameters. Okay, I can just tell you for this zero, obviously you need to add zero of the stock solution, and this one you need to add 1 ml. Sorry, it should be 10 ml because you are preparing 10 ml of total volume. Okay, so I hope you know how to calculate the rest. Okay, so once you have done, it says here you take 3 ml only. Although you have prepared 10 ml, you take 3 and maybe you need to repeat. You can use the other 7 ml later. But if not, just take 3 ml and then all the 6 test tubes, you repeat steps 8 to 11. Same thing, add 3 ml DNS, heat it for 5 minutes, and then add the Rochelle salt while still warm, and then cool down, and then finally read the intensity of all the concentration to find out what are their respective absorbance. And finally, you are required to make a graph of absorbance versus concentration for the glucose, not the unknown. Although you have three test tube for the unknown, but only the glucose will be used to make the standard curve. And by using the standard curve, you can then use to calculate the concentration of your reducing sugar found in your banana preparations. And after that, you answer this question or task. So I want you to go and find out what is the brand of the micropipette we use in our laboratory, as well as spectrophotometer, and go and search good YouTube videos, or now TikTok as well, and then submit the URL to me. So that is a way of forced learning for you, and also with evidence that you already learned what is in the video. So, why is the Rochelle salt solution required to be added while the tube is still warm? So, answer that as well. And is there a way for you to give me a suggestion how to cool, to room temperature, the test tubes without, just waiting it out for a long time on the bench? So, find out and also, or suggest to me. So, next one. Why is it necessary to neutralize the sample to pH 7? So let me know. And then this important thing that you will see in biochemistry, what is blank as well as control? So answer that as well. There is a huge difference between blank and control, but keep on saying when I mark your senior's practical report, it's a bit inaccurate in terms of both of the terminology between blank and control. So find out accurately what is the difference. Okay. And that's it. I have concluded the briefing for this practical, which is the practical of estimation of reducing sugar found in banana by dinitrosalicylic acid method. Thank you.